Thank you. So this is Dr. Martin Howe. He is with the University of Bristol. He's working in the ecology of vision group within the university and is interested in invertebrate visual systems and maybe how learning about visual system can help building better biomimetic machines.
Thank you. Yes, some biological soft robots. I'm fascinated by some of the robotics work going on with soft robots here, and it's really nice to see the parallels in the animals that we're looking at. And hopefully there'll be some inspiration for some of your work here. This is work done mostly by postdocs and master's students in Bristol, as well as Nick Scott Samuel in psychology. And we've also got collaborators at the University of Queensland and University of Cambridge who are working on a lot of this work. So I'm going to talk about two projects that we've been doing in recent times, looking at cephalopods, particularly cuttlefish. The first one is looking at how they deal with dynamic illumination. And the second is looking at how there's a species that mesmerizes its prey. But I'm going to launch straight in because time is short. This is the sort of environment that a lot of cuttlefish live in. So they live in these very shallow water environments, and you get this very, very tricky visual dynamic illumination going on. So how these animals perform normal everyday tasks in this sort of environment has been of interest to us. This visual noise in shallow water comes as a simulation that we use in a 3D simulation environment. But essentially, it comes from surface waves lensing light from the sun, causing them to converge and diverge on the sea floor. So you get this dynamic illumination, which depends on the surface wave properties, depends on how much sun is visible, depends on depth and turbidity. And throughout this, I'm going to call this caustics, or dynamic caustics. People love simulating these in computer game environments, and we actually take advantage of that in our experiments. But first, there's data to show that a lot of marine animals and freshwater animals are negatively impacted by this dynamic noise. So here, these are sticklebacks, and they tend to avoid areas that have these dynamic illumination. And they tend to swim at different speeds. They spend less time stationary when there's lots of this dynamic illumination. Also, it affects their ability to detect predators. This is a study by Sam Machette. He's trained these triggerfish to find targets on a touch screen in different dynamic illumination environments. And he found that almost every time that background had dynamic illumination, they took longer to find prey. So it's harder to find a target in this sort of environment. Now, there's a theory that perhaps this might be one of the original drivers for color vision in animals. Here's a scene of the early Cambrian seas, and the artist has conveniently rendered a dappled illumination in the background. And this is the environment that eyes originally evolved in, the complex visual systems that we see today. And this guy, Vadim Maximov, came up with the suggestion that maybe this is one of the drivers for evolution of color vision originally. And this is because caustics, this dynamic light, has no color component. It's an illumination, it's an intensity modulation over time. So they're achromatic. And that means that if you have color vision, the relative activity of two of your color channels is going to stay the same. So let's imagine you're filming this with a camera. The blue and the green channel of the camera will be fluctuating up and down, but the relative activity will stay the same. So if you compare the activity of those two channels, you might be able to filter out that dynamic illumination. There's very little evidence for this actually happening. There's a few modeling papers that look at it. But it could be one of the early drivers for color vision in animals. We thought, our lab works on polarization, so we thought, well, actually the same could apply to polarization vision. So the necessary prerequisites, caustics need to vary in intensity, but not in polarization. The animals need to be able to see the polarization of light. They need to have at least two channels of sensitivity to polarization. And also the things that they're interested in need to have a contrast in polarization with the background. Now, many of you engineers and physicists will know a lot more about polarization of light than me. But just as a very brief overview, when I'm talking about the polarization of light, I'm talking about the angle that the photons are making as they travel through space. So here we have unpolarized light passing through either a horizontal polarized filter or a vertical one, which is only allowing one orientation of polarization to go through. And the way that we measure that is at the moment using some nice camera sensors that are available now, which are, instead of having red, green and blue subpixels, we have subpixels with different polarization filters overlaying them. And that allows us to calculate, based on the differential activity of these subpixels, it allows us to calculate the degree of polarization within our images. If you'd like to talk to me more about the sensors that we use, there's lots of information online on this website in particular. So we took our cameras underwater and we asked, are caustic patterns underwater, as we look at the seafloor, do they have any polarization components? So here on the left, you've got just a standard intensity image. That's an average of all of our 4 polarization channels. And here we've got the percent polarization, or the degree of polarization, as a heat map with this scale here on the right. And hopefully you can see there's not much polarization there. It's all very, very low level polarization. And there aren't much in the way of fluctuations that correspond with the fluctuations we're seeing in intensity. Just to show that in more graphical demonstration. So here are 9 points on this video, and we've plotted over time the fluctuations in intensity. And then below that, the fluctuations in the degree of polarization. You can see there's very little variation in polarization, certainly not that corresponds with variation in intensity. So that suggests that these modulations that you get in the natural environment, they vary in intensity, but they don't vary in polarization, which is our first prerequisite satisfied. What about polarization vision? Well, we're used to seeing the world in color, but we look at crabs in our experiments, and they have this beautiful architecture in their eyes, giving them 2 channels of sensitivity. One bunch of cells that are sensitive to horizontally polarized light, one sensitive to vertical. And we've done behavioral experiments using modified LCD screens where we show them polarization cues. We found really strong reactions to very, very small contrast in polarization, as small as 3 degrees in angle between an object and a background, or as small as about 8% polarization. So they're very, very sensitive to the polarization of light. And the same is true for cuttlefish. Now, these animals have no color vision. They're completely monochromatic, but they are very, very sensitive to the polarization of light. So here we've got the log of the angular difference in polarization between an object and a background. We're getting responses as small as 1 degree in angle difference between an object and a background. So very, very sensitive. Now, this is for a specific visual task. These are animals that we're showing a predator-like stimulus. So they're getting startled by this predator that's in polarization. If we do the same experiment for a different visual task, this is a stabilization. So a bit like a stabilization rig on a camera system, our eyes and the eyes of crabs, they need to try and keep the world stable around them. So if you rotate the world around a crab, it will try and track it with its eyes. What you're seeing here is a crab inside an arena with a rotating pattern, and you can see its eyes are tracking that pattern. And this is the plot of the eye angle over time. And you can see they track it and then they snap back, track it and snap back. And they do this really effectively when you show them a black and white pattern spinning. And you can get a nice plot of how that varies with the spatial frequency and contrast of the pattern you show. If you do this in polarization, you get absolutely no response. So these animals, we know they can see polarization because we've shown it to them as a predator stimulus, but they ignore it completely when it comes to eye stabilization. So visual systems of animals aren't one size fits all. They're very specific to the task that they're doing. This task is using a certain subset of the visual inputs that their eyes are receiving and ignoring others. Does polarization mitigate visual noise? Coming back to our original question. Well, we use this standard setup in our lab where we stick a crab on a treadmill. I'll show you some videos later. And then you show it very carefully calibrated predator-like stimuli, and you look for a response. And in this instance, we've got one projector giving us a caustics background and another projector giving us an intensity-based predator that they're responding to. This is the kind of thing that they respond to. Let me just see if I can press play. So here's the kind of cue we show them. Simple expanding disc. Really, really nice and simple, but they really don't like it because it looks like an approaching fish. We also just slightly modified the setup for a second experiment where instead of having 2 intensity projectors, we have just a normal intensity projector, but a polarized screen. That's a modified computer monitor where we've taken off the polarizers. And that lets us present a predator in polarization instead. So you have an intensity background with a polarization predator. This is what the crab looks like on a treadmill. Poor thing, they get returned to the beach afterwards, so no crabs are hurt in this experiment. And this is our arena. And then we have two different treatments of background, a static and a dynamic illumination background. We want to see whether there's an effect of this sort of signal. This is what the crabs do when they're startled. There it is, just walking on its treadmill. And there you see a really nice, clear response. So they either freeze or they stick their claws up. It's a kind of a real predator aversion. Same sort of thing that happens when you approach them on the beach. So here are results. So what happens when you show them intensity-based predators? So this is the contrast of the predator and the response probability. As the contrast of the predator increases, so does the probability of responding. So a really dark predator against the background, they respond very, very strongly. When it's very low contrast, they don't respond so well. And that's a standard sort of sigmoidal response curve that we get for crabs. If we have a dynamic background, they're much less able to detect that predator in intensity. So in this context, they don't do anywhere near as well when they're trying to see that predator. So that visual noise is diluting their ability to detect the predator. What about in polarization? So here we've got now the stimulus is in polarization against these two backgrounds. And as the polarization contrast increases, so does the probability of responding. So they're really keying in well to this polarization stimulus. And in the dynamic background, actually they do just as well. So the polarization, when the predator is viewed in polarization, they can do it equally well in this noisy versus static environment. So that really strongly suggests to us that they're using polarization to get around this visual noise problem. And we weren't content to stick with crabs, so we then moved on to cuttlefish, which are clearly the best animals in the world. Here we've just modified it, where we've got the screen is presented on the front of aquaria. And this is what the cuttlefish do when they're startled. See a nice little twitch of the body. And sometimes you'll see changes in color in the chromatophores. There's a very subtle response there. And then another one here. So you get nice, clear responses, but a little bit more subtle than the crabs. And here's the data presented in the same way. So for intensity, again, they really didn't do very well when the predator was in a dynamic background. And for polarization, they did equally well. Now, the data size wasn't as big for this experiment because it's much harder to do this kind of thing with cuttlefish. But those two animals combined, we think it's pretty good evidence, therefore, that polarization vision is mitigating the negative impacts of dynamic noise underwater. So we're keen to take this now into natural settings and to extend it into chromatic contrast, like the Maximov theory, but also to investigate ways of modeling this sort of thing in silico. So, you know, how motion vision works in animals would be really cool to try and apply that to camera systems and processing algorithms to see if we can find some cool uses for this kind of thing. So the second experiment I wanted to talk to you about was a cuttlefish experiment we embarked on recently with this species, the broad-club cuttlefish. Now, it's a really charismatic species that you find in tropical oceans around the world. And we were interested in this really interesting behavior that they have when they're hunting using repetitive moving stripes. Repetitive stripes have been used quite a lot in past for attempts to camouflage. This is during the World Wars. A guy called Norman Wilkinson came up with the idea of dazzle camouflage as a way to protect ships from enemy attack. There's also this barber pole illusion, is an illusion that confuses our visual systems. This is a horizontally rotating cylinder with diagonal stripes that look like they're moving upwards. And high contrast markings are really widespread in the animal kingdom. We were quite keen to see whether maybe any of these optical illusions might be behind the evolution of things like swallowtail butterfly wings, or this is what a beautiful lionfish that you get out in the reefs, and of course the iconic zebras. We'd spent a lot of time trying to find out whether these high contrast stripes, which are used on kind of all sorts of optical illusions, whether they actually do anything to visual systems in nature. And there's a whole bunch of experiments, some of which supporting the idea of motion dazzle, and some refuting it, either through human psychophysics experiments or attempts to try and model visual systems. But some of the most unique dazzle displays, I think, in nature are in cuttlefish, partly because they've got this ability to change colour so easily. Here's a beautiful flamboyant cuttlefish. This has got this warning coloration. You can see these moving bars of colour that it passes across its body. So really remarkable. And then we come to this animal, the broadcloud cuttlefish, really special animal. And it's got a very unique hunting technique that some of you might have come across if you've watched the Blue Planet 2 series recently. So this is a series of frame grabs from a hunting cuttlefish. Now there's a crab just at the bottom of the field of view, and you can see it sort of takes a very strange posture where it spreads some of its arms out and it passes stripes across its head. So we set out to try and film this using a very basic stereo camera rig. This is 2 GoPros using a camera calibration toolbox in MATLAB to then be able to recreate in 3 dimensions what's going on. And we're able to film this in nice high resolution at nice high temporal frame rates as well, so because some of this is quite fast moving. And some very early data off the press, we've got some nice videos of this recently. Here's an example. We've got a cuttlefish here. There's a little prey item here. And this is what the cuttlefish does when it sees this crab. Sort of approaches in camouflage and then sticks out its arms and passes flashes of color across its face. Really remarkable. Here's another example, a slightly slower one. And there's a lot of individual variation here. So this is a different individual, but still in real time. There's a crab just at the bottom of the screen here. You can see passing these amazing pulses of color across its head as it's coming into strike. And we're fascinated by why the animal's doing it. Is it some sort of dazzle display? Is it mesmerizing the prey, as they call it? So we've embarked on an attempt to try and get to the bottom of this. First thing we wanted to ask is prey behavior altered by the moving stripe pattern? And we went back to our favorite treadmill experiment, crab on a ball, and we show it different approaching predators with different stripe patterns on them. And this is the kind of thing that we're showing them. So we're showing them 3 different treatments: a gray, static stripes, and moving stripes. And these are all sort of looming objects with these different properties. And again, we have the type of reaction that we get is very similar to in the last experiment, where you have the approaching object and the crab, in this instance, stops walking. And in this instance, it'll stick its claws up in an attempt to defend itself. Tends to be the big, well-defended males that do that. So we asked, does the stripe display have any effect on the ability of crabs to detect these approaching predators? So here what we've got is just the grey stimulus. So this is when we show them grey in different levels of contrast. You can see the probability that the cuttlefish, that the crab responds is increasing. The same for a static stripe. It looks very, very similar to this. So as the contrast increases, so does the probability of responding. But when we have moving stripes, we find that something very weird goes on. So at the very high contrasts, the probability that the crab responds seems to be a little bit lower. So there's definitely something interesting going on here. There doesn't seem to be anything in it when we look at the timing of it. So they seem to be responding at more or less the same time as this thing is expanding. But there might be some differences in post-detection behavior. So here we've got 3 different behaviors: raising claws, running, or tucking claws. And they seem to be less likely to show an aggressive response to this moving stripe stimulus. But they're not much difference in these other 2 behaviors. So there's some interesting, that's sort of the first experiment that we've set up on to try and understand this. We also tried to use some motion simulations to try and work out what this might look like, what the display might look like to the crabs. And we've been using these very basic elementary motion detector models, which are sort of modeled on a biological motion detector. And essentially what we get is a map of where the motion is detected in a scene. And we just compared like an expanding noise image with a hunting broad club cuttlefish. And so an expanding noise image, we get sort of a nice expanding motion scene. This is color coded according to this border here, where you see sort of the world just expanding in a predictable way. And that's a polar histogram of the motion. If we look at the cuttlefish, though, when it's approaching, we actually get a very strong downward moving component to this, which shows up in this polar histogram of the motion cues. So when we simulate it from the perspective of a crab, it could be that actually what's happening is this downward motion is overloading the visual system and overriding any weak expanding signal that's there. So it might be a motion noise mechanism. So we're keen to do more crab experiments to really describe the effects of the display across the stimulus space. We want to look at natural hunting behavior to see whether it's optimized for crab vision. We'd also like to look at the really fine scale context that they do this hunting behavior. And we're also keen to reconstruct these hunting sequences from our stereo video sequences to get the three-dimensional interactions and maybe recreate the field of view of the crab. But one of the things that we then get from this is nice three-dimensional information of these sort of soft robots going about this very complicated hunting behavior, which could be of interest to some of the roboticists here. So thank you very much. And yeah, I'd be lovely to have some questions.